Yeah. Can you see the presentation?
Yes, it's clear.
Okay. So as I said, more than the subject, bioinformatics is a tool set. It's a toolkit. There are very few sub-disciplines within the botany or biological sciences which make you a professional. And bioinformatics is one subject which makes you a professional in a way. So that way bioinformatics stands out in comparison to all. Just one second. That was better. Okay, can you still see the presentation?
Yes, Dr. Suman.
Okay. So the first thing is just to warm up to the topic. Why bioinformatics? There's a very interesting data I have that, until 2003, from 5,000 years from then backwards, the humanity had created a total of 5 exabytes. I can't even explain how much that is, but trust me, it's a lot. From 2003 to 2010, this amount, which was created earlier, taking 5,000 years, this amount of data was created every 2 days. And it's believed that from 2010 to 2013, this very amount of data or information is being created every 10 minutes. And presently, within hours, all the information which has existed, it is exceeding, on hourly basis. So literally, there is an explosion of information. That is what we have in the public domain, mostly in the public domain. So when I say information, which is again, information is an integral part of the subject which you have been deliberating in the last 7 days. What is information? Simply said, information is that which informs, or in other words, it's an answer to some kind of a question. So when it comes to bioinformation, it's essentially an information about living organisms, or in other words, it's an answer to a biological question of some kind. Why information? Why do we need it? Information reduces uncertainty. And why so much information? Information is important. We said it reduces uncertainty. But why do we need so much information? They say there is a relationship. More uncertain an event is, more complex a phenomenon is, more information is required to comprehend it. Life being so complex, so intricate, and so unpredictable, we need huge information to resolve its mysteries. So we have been emphasizing that information is important. Where does information take us? What do we do with the information? Information takes us to knowledge. So the precursor of knowledge is information.
Sorry for the interruption, ma'am.
Yeah.
The slide is not moving. If you are
okay. Slide moving a bit.
We are
just give me a minute. I'll have to restart. Yeah.
Ma'am, you can have the slide show from the bottom toolbar.
Yeah. It's better. Is it okay now?
No, ma'am, you haven't pushed the slideshow button. This one, yes, this one.
Is it okay now?
Not this one, ma'am. एक और cursor उधर लेके जाइए. वैसे it is moving.
Slides are moving now. So should I be in the slideshow mode or in the edit mode?
It can be either ways. Slideshow mode will be better, I think.
Achha. So.
Yeah, this one.
This is okay now? Can you see it?
Yes, ma'am, we can see. It's okay, ma'am. इसी उसमें आप.
Ma'am, please click. You can see the battery percentage 75. The icon next to it. Please click on that.
I'm in the slideshow mode, so I can't see it. Okay, no, I am.
Okay,
No.
okay.
Yes, ma'am.
this
This
only.
this is the
This
one.
only.
This is the
Yeah,
one.
I'm using that only. I'm using the
Acha,
slide.
but it is not visible here. It is visible as the
Edit
slide.
mode. I know this happens. I don't know why.
Okay, ma'am. Okay, ma'am.
Okay,
It's fine. It's
okay,
fine. The
okay.
slides are moving.
Okay. So please tell me whenever there is a problem. So
Yeah, sure.
I was telling that, information being an integral part of the term bioinformatics, I wanted to spend some time on what exactly do we mean by information and where does it take us to. Information takes us to knowledge. And why do we need knowledge? We need knowledge for our own survival, for our basic survival, whether it is food or medicine or protection from natural or man-made calamities or a better life. I'm not even going to the philosophical aspects of it, that it tells us the meaning of our existence or sense of purpose, but the very survival of mankind on this planet needs knowledge. And a very interesting information I would like to share with the students, particularly here. Donald Rumsfeld, who was US Secretary of Defense, he was talking about a very different aspect. He was talking about foreign entanglements in US and its consequences. He has given a very interesting classification of knowledge, all the knowledge. He says there are known knowns. There are things that we know them. And there are known unknowns. That means we know that there are certain things about which we have no idea. We do not know about them. And then there are unknown unknowns. Things we don't even know that we don't know. This is the most interesting part of it. Easy to comprehend for this student participants is that, when you are in the school, you have learned biology, you have learned photosynthesis, you think I'm sure all of you must have been good students, you thought you know photosynthesis, and then when you come to graduation, you learn totally different aspects of photosynthesis which you had not even heard of in your schooling. Not only students, even as scientific community, let me tell you, till 1993, we had not heard of microRNAs. We knew so much about biology. We had explained so many biological phenomena and processes without even knowing that there is a small non-coding RNA, which now we believe has played or does play a very important role in almost every aspect of biological, world. We didn't know that they exist. So the whole of the science or whole of research is basically either moving from unknown unknowns to the known knowns, or some people believe the journey is backwards, that we move from known knowns to the unknown unknowns through intermediate steps of known unknown. That means we try to identify what are the gaps in our existing knowledge, and that is what actually makes the world more knowledgeable. And when it comes to scientific knowledge, essentially scientific knowledge is about the physical world, a phenomena which is, scientific knowledge is always acquired using scientific methods. That means there has to be a logic, there has to be reasoning, there has to be evidence, there has to be observable features. Only then, it is accepted as scientific knowledge, because not only knowledge, we are talking about scientific knowledge here. So if information is precursor of knowledge, where does knowledge takes us? Knowledge takes us to wisdom. So this is how the chain goes. Precursor of wisdom is knowledge. Precursor of knowledge is information. And what is the precursor of information? Where does information come from? Information comes from data, the scientific data, collected scientifically, collected meticulously. And this is where bioinformatics comes into the picture. Because nowadays we are in big biological data era. There is too much of data, which is the precursor of information, which becomes the precursor of knowledge. And it is tools like bioinformatics which are instrumental in comprehending that big biological data into useful information, which with human intervention becomes knowledge and subsequently to a wiser mankind. This is the importance of bioinformatics, particularly in the present era when we have huge, big biological data available in every sphere of biological sciences. That's the importance of bioinformatics. And that is done considering the limitations of human brain. It is absolutely aided with the help of computers or computation. When you talk about data, now I come to the very, practical considerations for the students who want to enter into the field of bioinformatics. Even if you are just initiating, these may seem very trivial things, they may seem very trite, but remember, these are the, you know, my experiences, or these are the experiences of many people like us who have learned bioinformatics later, or who have at least tried to use bioinformatics in their existing research areas. When I said that all of the information will actually come from the data, when you
Sorry
are
for
doing,
the interruption, ma'am.
again,
Your
you
slide
missed out?
is not moving. We are still on slide 5, ma'am.
Slide
And
maker,
it
1
could be
minute.
the reason that when you were sharing your screen, you have shared the PowerPoint presentation. If you share the window screen, that will be helpful.
Okay, 1 minute. Just give me 1 minute, Dishan. Window screen share. Window screen share.
Stop sharing.
I can stop sharing the paper. This thing here.
May I suggest the solution, ma'am?
Yeah, no, I'll put it on. Okay, yeah, tell me.
If you can click
Click what?
Control Shift and E. Control Shift E.
Shift E. Okay.
Control Shift E, and then you get the option of screen. After you click
No,
Control Shift.
let me. Just 1 minute. Just 1 minute. Is this Dr. Jishan speaking? Jishan?
Yes, ma'am.
Yeah. Okay. Okay. So now I have it in the edit mode, PowerPoint presentation. Can you see that? No.
No, ma'am. It's not visible.
Okay. Okay. Now you tell me what should I do, because I had come out of it. That's why I came back.
Okay,
Now tell
no
me what
worries,
should I do.
ma'am. You can click Control plus Shift plus E,
Okay.
and when you do it, you get the option of share content.
No, I'm not getting.
Or
Dr. Suman, Lakhanpaul, sorry, Dr. Zeshan, why don't you give us the presentation and we'll present it for you?
Okay.
Can you mail it to Dr. Tabassum or Dr. Ruchivir, whomever you
Okay.
have
Okay. Okay.
access?
Okay. Okay.
Just send it and they'll do it.
Okay. Okay. 1 minute. Just presentation. Okay, then mail. Yeah, mail.
I have to say me. Compose.
मेल करना है उनको presentation will switch to the speaker.
Professor Suman,
I'm
till
doing
you
it.
do
I'm doing
this,
it. Yeah.
we
Please go
just
ahead.
invite somebody to give oral feedback. Is that okay?
Okay, please go ahead. Yeah. Please go ahead. No problem.
So somebody by the name Vinay had asked for oral feedback. So Vinay, are you there? Can you raise your hand? Vinay from Ramjas College. Yeah. Dr. Ruchi, can we have him as a presenter? He'll quickly.
Yes, ma'am. Yes, ma'am. Yes, ma'am. So, Mr. Vinay Verma is a presenter now. So he can switch on his camera and mic and he can speak.
Good morning, ma'am. Am I audible?
Yeah, you are.
Thank you, ma'am. Good morning to one and all present here, all the esteemed presenters, all the respected researchers, and mostly my heartily touched one, all the participants who are sitting here and studying. First of all, I'd like to thank Zakir Hussain College for allowing me to join this esteemed workshop with all the esteemed presenters. And I'd like to thank all the conveners and all the organizing team for providing us a comfortable environment to enjoy and study at the same time. The practical part I felt is most heart-touching because in most of the workshop I felt that theory is taught and practical is not conducted, which seemed to be quite boring. But here we can ourselves do the practicals under the under the guidance of our esteemed presenters. And I like to thank all the presenters to inculcate about this new field bioinformatics. All I know about bioinformatics is just the blessing of this workshop, and I thank you all for allowing me to speak and being a patient listener. Thank you, everyone. Thank you again.
Thank you, Vinay. We have Tanmay Malik from SGTB Khalsa College who wishes to express regarding the workshop. Tanmay?
Tanmay can raise his hand so that I can make him presenter.
Hello.
Thanks organizing team of Zakir Hussain Zakir Hussain Delhi College for organizing such a wonderful and amazing session. It was really great to be part of it. Further, we learned and explored a lot about the bioinformatics that is gonna be that is gonna to be applied field in biology in the next 20 and 25 years.
Thank you, Tanmay. And thank you, Vinay. Anyone else wishes to give oral feedback till Professor Suman Lakhanpaul is back? You can just raise your hand in case you are willing to speak. Yes, who's that?
It is Gargi. I am making her presenter now.
Gargi is from which college?
Ma'am, it's not mentioned here. Maybe she can introduce herself also.
Yes, ma'am. Thank you so much, ma'am. Good morning, ma'am. Good morning to all of you here. I'm Gargi from Sri Venkateshwara College, and I'm so happy to know about bioinformatics. As such, I was not so much interested in it, but as I learned from the workshop, I understood its importance and how we can solve our questions regarding so many things. And indeed, it was so happy feeling, Abhi. Thank you so much.
Thank you, Gargi. Anyone else who would like to speak? Please raise your hand in case you want to speak. And Dr. Ruchi, let me know as soon as Dr. Lakhanpaul is back.
Yes, ma'am. Ma'am, there is a attendee by the name of Anmol. He also wants to speak. So I am making him presenter. And yes, Anmol is a presenter now. He can introduce himself and he can give us the feedback.
Good morning, ma'am. Am I audible?
Yes, you are. Good morning, Anmol.
Good morning to everyone, all the resource persons, organizing team, and the participants. My name is Anmol. I'm from Bhaskaracharya College of Applied Sciences. I have recently graduated from zoology honors and studying bioinformatics as an extra course here, as an extra in a workshop, is a very is a cherry to take because in our curriculum we all see that we just get a gist of bioinformatics and the immunoinformatics, if I say. And regarding these seminars and workshops which different colleges and different departments organize, actually give us the opportunity to learn a lot of new stuff and new things coming up in the future, because we know we live in a sci-fi tech world and we need these stuffs to these things to upgrade our knowledge and upgrade our research things. So thank you so much to the organizing team of Zakir Hussain College for giving us the huge number of participants a chance to be the part of the discussion. Thank you so much, ma'am. We really loved it, and I am looking forward for future seminars and workshops too. Thank you, ma'am.
Thank you,
Thank you.
Anmol. I think Professor Suman Lakhanpaul is back. So can we go back to the screen?
Ma'am, I think ma'am will take, Dr. Suman will take some more. Ma'am, are you, Suman ma'am, are you
I'm
ready?
there. I'm there.
Yeah,
The
hello.
Yes.
presentation
Yes,
is
Yes.
Suman ma'am is there. Dr. Suman Lakhanpaul is there, and Dr. Zishan. I'll be projecting her slides, so kindly see that.
so I'll be requesting Tabassum you to forward the slides.
Just projecting.
Okay.
So is it there?
No, not yet. Yeah.
Just a minute, ma'am.
Yeah, it's there now.
Is it there?
Yeah,
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 yeah.
yeah.
Yeah, it's there. It's there.
Let me. Yeah, is it there now?
Yes.
Yes.
Okay. Thank you.
So Dr. Jishan told me that I was at slide 5 because for me they were moving.
Just a minute, ma'am.
Fine.
This one?
Yeah. Okay.
Yeah.
Okay. So is it okay? This is where I was. Everybody feels that? Yeah. Okay. Okay. So I was, since information is an integral part of the term bioinformatics, I was just trying to emphasize on the word information, what exactly we mean by information. Next, please. And why do we need information? So can I? Will I be able to forward it? No. Next, please. Yeah. So why do we need information? We need information for knowledge, because information is the precursor of the knowledge. Next, please. And I said that knowledge, very interestingly, has been classified into 3 categories: things that we know them, and things, oh, it is gone.
No, it is there. It is there.
Oh, I have lost. Okay, fine. So, and there are things which we know that we do not know them, and there are things which are not known that we don't even know about them. And a very good example of that, I said, is the small non-coding RNAs for the scientific community, which for us till 1993 did not exist, and they have been proved to be so important. Similarly, a subject like bioinformatics, we didn't know that it is such an important subject, and now it has become such a fast emerging and a helpful tool for all the scientific community now. Next, please. And the whole journey of science or research is, either moving from unknown unknowns to the known unknowns, or from known knowns to the unknown unknowns. That is how this whole scientific journey actually happens. Next. And when we say knowledge for which the precursor is the information, we are essentially only talking about the scientific knowledge which comes from the physical world, and the physical world understood using scientific method of inquiry, using observable and measurable evidences derived with the help of proper reasoning and experimentation. Next. So if we say that information is the precursor of knowledge, then the precursor of wisdom is precursor for knowledge is information. Similarly, knowledge becomes the precursor for wisdom. Next, please. And all this chain of information, knowledge, and wisdom actually comes from data, the scientific data. Next, and that is where next, you can skip 1 or 2 slides. I think I have talked about it. Next. So this is where I want to emphasize that since this is an era of big biological data, which is very challenging for human mind, it is the help of computers or the computational ability that bioinformatics has come into playing a very important role to transform this big biological data into information, or with the help of human intervention, from information to the knowledge. Next. Okay, so this is where I think I will address some of the very practical considerations of a working scientist, whether he's initiating into bioinformatics or he's already a practicing person using the bioinformatic tools. When we say that the whole thing is, on the foundation of data, when we do bioinformatics or we use bioinformatic analysis in our research, there are 2 kinds of data we are handling. Either we are handling primary data or we are handling secondary data. Primary data, when we say, that means we have done experiments in the lab, we have generated some results, some raw data, and that is what we are now subjecting to bioinformatic analysis. So these are the advices, these are the words of caution from a senior person. Remember that the data which you use for bioinformatic analysis should be extremely accurate and reproducible. There is no point drawing very big conclusions using big softwares where the data is not very reliable, or what we call a fuzzy data. That's a very important thing. Secondly, you should also know that much of the raw data which is obtained using experiments in the lab has to be submitted in databases. Research publications in the field of bioinformatics or in the biological sciences are not acceptable if the data we have obtained, primary data we have obtained, is not submitted in some authentic databases. Second is the secondary data, where we use for bioinformatic analysis many of the input data from databases. There are so many databases, and I believe that you have been very nicely informed about the databases. So many a times we use data from secondary databases. It is as important to test, to check the accuracy and the quality of data which we take from secondary databases, again, because we have not generated it ourselves. They have been submitted by somebody, and we are not sure about its quality, not only in terms of, say, if there are DNA sequences, we are not sure how rigorously those sequences were obtained. In fact, to the best of my knowledge, even today, NCBI does not insist on submission of chromatograms. There are some databases which insist that when you are submitting some sequences, we should also deposit the chromatograms. But not many of the most reputed databases are still doing that. Second thing is, whenever we are doing bioinformatics, we are essentially dealing with some data from some living organism. The taxonomic identity of the information which has been deposited in the databases which we are going to use it, we are not sure whether it is absolutely correct or not. This is again a very important caution which you all should use when you do bioinformatics. Next, please. I'll just give you an example here. These are, examples of chromatograms obtained by Sanger sequencing. The bottom panel shows the ideal chromatogram of a DNA sequence. So if we are generating this sequence by our own lab experiments, we have done some amplicon generation and got the sequencing done. If I have got this kind of a panel, I am very happy that I have got a very good data. But very often we do get results like this, which are bad chromatograms. There are regions, not the complete chromatogram is bad, but there is a region in the DNA sequence which is not good, something like this or something like this here. As you see, we will get some DNA sequence, and if that sequence is submitted in the secondary database, if it is from my own lab, I know that there was a patch which was not a very good sequence. But if I'm using a secondary data, I am not sure how much of it is actually very, very reliable. And secondly, as I said, the taxonomic identity, there is no way for a database to know the person who submitted the sequences, did he really identify the taxon, whether it was a bacteria or a plant or an animal, actually in the most correct way. And if we use those data points or those sequences, whether it is an amino acid sequence or a protein sequence, blindly without knowing whether it was really correct identity or not, we will be in trouble and we will be drawing wrong conclusions. Next, please. Another word of caution I would tell to the students is that just like we try to propose a hypothesis in the wet lab experimentation, when you do bioinformatic analysis, try to pose a good question, ask a good question. It is very easy. You take any data, you subject it to a software, you do bioinformatic analysis, you will get some results, some output will come. But what is it that you were actually trying to answer? So normally in wet lab experiments, we are very careful about it. But when we do bioinformatics, we have to be equally following the same procedure. Before you start a bioinformatic analysis, ask a good question. Keep in your mind what is the question you are trying to answer. Next, please. Next. Okay. No, there is, I think you have 2 slides you have gone ahead. One more. One, I think back you have to go. Still one more back. One more back. Yeah. So next, next, please. Yeah. So this is, now again you have slipped one more slide. Just one we had to go. You have skipped one more. One slide back you have to go, Tabassum. Hello, yeah. After that, one more slide. Next slide. Yes. So this is as far as I told you that before you start with the analysis, you have to be careful about the data. You have to be careful about what question you are doing, answering. While doing bioinformatics, again, as far as possible, try to use the most recent versions of the software. It is not always possible. I know there are practical considerations, but this is one issue which comes up when we send our publications. They'll say that, okay, when Mega 11 has come, why are you using Mega 8? So this is, as to a great extent, we should be very careful to keep a close track of which is the latest version of a particular software you're using. Another advice I will give to all researchers or all students who want to use bioinformatics is, whenever there is a software which comes, very frequently or very often, there is a research paper which accompanies that software. That research paper gives you all the important points, all the premise of that software. What are the important features of that software? What are the do's and don'ts of that software? Not only for each software, every time there is a newer version of a software, there is a research publication. So we should try to not only read that paper, but very frequently we should refer to that paper. Every time we will encounter something or other, the answers to which will be given in that original paper. Never, those things will never be addressed later. A very important observation I have with the students is that one minute. Any software comes with the default parameters. I've seen students don't change those default parameters. They keep on doing analysis without altering the default parameters. Every parameter has some alternate values. Try to play around with that. By playing around with the parameters, you will be making your analysis either less stringent or more stringent. And you will really get to know a lot about your data, about your work. Yet another thing is that try to use the same data set, asking the same question with different softwares. That will tell you about the robustness of your conclusions. Don't depend only on one software. When we are trying to answer a question, keeping the same data, keeping the same question in your mind, try to answer the same question using different softwares. There are always some differences. Some parameters are different, some assumptions are different, and see how many times your conclusions are the same, or in what respects your conclusions are varying with the different softwares. That will make you aware not only about your result, about your data, but also about the features of the softwares, how the softwares are different, which have been claimed to be for the same purpose. Next please. So my request to the students is that do not use softwares like black boxes. You put in some data and out comes some result. Know what is happening, and to know what is happening inside that box is best ways is to change the parameters, intentionally sometimes change the data sets, and confirm the results using more than one software to know what exactly is happening with your experiment or with your research. Next. Again, very seemingly trivial thing, but very important that you will know only after some time if nobody advises you on it. Try to give unique IDs to all the data sets which you are using and also to the outputs. Initially, you will be starting, you can give very generic labels like tree, phylogenetic tree, ML tree, parsimony, like that. No. After some time, in some months or in years, if you do not give unique ID to each of your data set and as well as to each of your result, you will be confused, you will be in a mess. So make it a practice. Make your own codes where just by looking at the label, you know which experiment, which data set this particular file is about. So that is something which you have to be developing as a habit from the very beginning itself. There are certain things, certain precautions which software also tells you, each of the software, that avoid using space, avoid using dots in the labels, keeping in view the number of characters which are allowed for each label. Those very basic things, very practical considerations you have to keep in mind. Next, please. Okay. Once you have done a bioinformatic analysis, for the acceptability of it, most of the times we need to validate our findings. If we are making any claim, more often than not, just doing a bioinformatic analysis and telling this is what, is not acceptable. It is becoming more and more unacceptable now. You have to confirm your claims with the wet lab experiments. Any inferences which you draw has to be supported with the help of wet lab. Very basic thing is, if you are predicting the structure of a protein, the structure may be plausible, but it is not necessarily that that structure is present in your system. So, in silico analysis, bioinformatic analysis can give you some inference, but for the acceptability of it, it has to be supported by actual experimentation in the lab. Sometimes it's vice versa. We do some experiments in the lab, wet lab, and we do some results. We get some results, and then we need bioinformatic analysis to confirm that this kind of a result is possible, theoretically speaking, considering all the parameters. So it can be opposite also, where we test the feasibility of a wet lab result using bioinformatics. Here I would like to give an emphasis on this point, that it's not all bioinformatic analysis or bioinformatic results can be validated using wet lab experiments. For example, lot of phylogenetic reconstruction, all the tree or the diversity analysis, phylogenetic reconstruction is done using bioinformatics, and there is no way to do the wet lab validation of it. In that case, you claim the robustness of your findings only by, as I told you, doing independent analysis of the same data using different methods, and by either claiming or by establishing the congruence of the results obtained using independent analytical methods. So that is where the validation can be justified if you are doing that kind of a bioinformatic analysis, which is not possible to be validated using wet lab experiments. And here I would like to say that is why either the students, you have to be capable of the wet lab experiments also, as well as the bioinformatic, or in future, those of you who plan to take bioinformatics as a research career, you should be in a team where some of you are very capable bioinformaticians, and the other members in the team are actually workers in the wet lab or doing actual experiments in the laboratory. So because mostly it is difficult for a person to be equally apt in both, the best is to have a combination, to be in a team where some people are bioinformatician and some people are wet lab experimenters. Next, please. Again, since you are just beginning, I believe that many of you are now quite excited and looking forward to going in this field. I will request that even if you are a biologist, even if you don't want to be a full-fledged bioinformatician, learn computer programming. Whether you are a user or a developer, there are some important languages like R, Perl, Python, Java, C or C++, at least one, or if possible more. Try to learn programming language. You will need to develop some in-house small scripts. Whatever softwares are available, many a times they do not serve our purpose. We need to have some very small questions which need to be answered. For that, if you are capable of making your own small in-house programs, they will be very useful in your career if you want to take a bioinformatics more seriously. Next, please. Okay. Towards the end, I would just say that there are some, again, those of you who are from biology background, even then, if you are thinking even to go for bioinformatics, you have to have some background of technical or some knowledge about computers on the technical side. There are open source, there are licensed softwares, there are downloadable softwares, or there are online softwares. For each one of them, the requirements of your system are different. If you are using a downloadable computer software, then what are the as I heard the last part of the talk by Dr. Brijesh Rathi, and somebody had questioned whether he is capable of using Schrodinger or not in his system. So this is something I am referring to in this slide. This was a very good question and very relevant, which all of you will have to face at some point of time, that which software you want to run, whether your system is capable of running that software or not. Nowadays, there are many online softwares. Even if you intend using online softwares for your analysis, in that case, internet connectivity, the speed is very important, and the stability of your internet. You could be doing an analysis, but if your internet connection is not good, even if you think I don't need this software, it is online software, I can use it, you need a very stable and a fast internet connectivity. So these are the technical considerations. I am saying it because many of the biology students are not very familiar with these things, but if you have to go in this field, you have to make yourself familiar with some aspects of computers, hardware, not only software, but the hardware also. Next, please. Yes. So this is where I'm almost coming to an end here. I don't want to unnecessarily take into the next speaker's time. But I like this quote by David Weinberg. He's a very famous author and technologist. Very beautifully, he says that next Darwin is more likely to be a data wonk than a naturalist. So what it means is that next big thing in biology is going to come from people who are dealing with lots and lots of data. That means who are the bioinformaticians. They will be the people who will be coming out with something, really as earth-shaking as Darwin had done, so many 2 centuries ago. Next, please. But here, next, a word of caution that despite a lot of hype about big data, about computation, about bioinformatics, remember that they are only aids to human thinking. They cannot replace human thinking. So your thoughts, your ideas, your questions are more important than the data or the information you have been working on. So they will become knowledge only if human intervention and very intelligent and beautiful questions are asked. Only then information becomes knowledge. Otherwise, they remain not only information, but they remain just data, which is in plenty in the public domain already in so many databases. So with these words, and advices to the future bioinformaticians. I once again would like to thank the organizing committee for giving me this opportunity. My highest appreciation for the organizers of this workshop, Dr. Lakhanpaul, Dr. Tabassum Jahan, Dr. Ruchi V, Dr. Jishan, whom I know have worked tirelessly for first coming up with this idea and then transforming into reality. I have been told that the senior colleagues were very, very supportive, very encouraging to the young colleagues who have organized this workshop. And finally, I once again congratulate the participants of the workshop and wish them all the best to their journey in life, as well as especially in the path of bioinformatics. And I am sure that this is just a first step of their long journey in this exciting area of science. And most importantly, once again, I would compliment and congratulate the faculty of Botany Department, Zakir Hussain College. And to them also, I would urge that let them also let it be just a beginning of many such workshops in future, and they will continue to organize such useful activities in future also. All the best to all of you. Thank you so much.
Thank you so much, ma'am, to give such a beautiful and practical approach in a nutshell to what is bioinformatics, why, where, and how we can use it. It could not have been more appropriate to end this workshop than to have a really practical approach on the subject as given by you. Thank you, ma'am.
Can I log out now? Yeah. Can I log out, Tabassum?
Ma'am, you can be there for some time. It will just end up in 10 minutes now.
Okay.
I would like to say a thing to Dr. Suman Lakhanpaul. Dr. Lakhanpaul, please stay on till we have a group photograph on screen. Thank you.
Okay. Now that we have come to the end of this eventful week of learning, as organizer of the workshop, it was indeed a very satisfying experience to instill some inspiration in the students. We got registration from more than 90 institutions and universities across the country. And within the Delhi University, we got registrations from 26 colleges. We got participations from practically all states in the country, like Gujarat, from Punjab, Tamil Nadu, Bihar, Uttar Pradesh, to name just a few. This was indeed very encouraging for all of us. Though today is the end of this workshop, but it must not be the end of the learning. I hope that students will nurture the interest that has been triggered by this workshop. Curiosity is more important than knowledge, because curiosity leads to to learn more. Never stop to be curious. Never stop questioning. This is the only way to learn constantly. Learning is not restricted to a time period. Rather, it's a lifelong process. It is very important to have scientific temper in you, the young minds. In the words of Einstein, if science has to grow, the younger generation, who are the future of the country, must take up the challenge. At this point of time, it's indeed my pleasure to state that we have launched an official channel of Department of Botany, Zakir Hussain, Delhi College. The purpose of this YouTube channel is to provide the latest academic endeavors of the department. This is an attempt to constantly bring learners abreast with the latest in the field of science and environment through seminars, conferences, webinars, and workshops. Please subscribe. It is a request to all the participants and everybody present here to please subscribe this channel and feel free to suggest topics for future deliberation. Please note that all the sessions of the present workshop, they are also available on this channel. May have, this is the uh, link to this official YouTube channel, Phytozaccharia is there, and we will share it in our to your Telegram group also, and to the speakers also. We will share this link. So, with these, it is my pleasure to convey thanks to everyone involved to make this workshop a big success. First, I convey my thanks to Professor Suman Lakhanpaul for taking time out of her busy schedule and address the students. Thank you, ma'am. I also thank all the speakers of the workshop who have shared their knowledge and expertise with us. A heartfelt thanks to our principal, Professor Masroor Ahmad Beg, for providing all the support and encouragement for this workshop from time to time. Thanks are also due for the webinar committee of the college, especially Dr. G.S. Tuteja and Dr. Dheeraj Kumar for their technical support in carrying out the workshop online and for the YouTube streaming. I also like to extend my thanks to Dr. Devender from Environment Science Department of the college for his constant support in the conduct of this workshop. A big thanks to all the participants without whom this workshop would not have been successful. They have been a very patient and keen learners. And a special thanks to all the student volunteers for their active participation to make this workshop a success. And at last, but not the least, thanks to the Department of Botany, Zakir Hussain Delhi College for the support and cooperation that was available during the whole time period of the workshop as well as before that. Thank you everyone and have a safe and great times ahead. Now I would request to everyone to please switch on the camera so that we can just have a group photograph. Thank you. This is just to remind to the participants that there's a feedback form that is posted on the Telegram group. All of you please fill that feedback form and submit to us as soon as possible, latest by 3 today. The filling up of the feedback form is mandatory to qualify you to get the certificates. So I once again thank all of you. Thank you, ma'am, for accepting our invitation and coming to address us. Thank you, everyone.
Now,
Dr. Tabassum?
Yes.
Can you tell about the statistics of workshop, the participants and the institutes they have joined?
I just mentioned that we have got the registration from more than 90 institutes all over India, and within Delhi University also we got participation from more than 26 colleges.
Dr. Tabassum, I just want to intervene that because the attendees are unable to switch on their camera, and our volunteers and staff members are also included in that. So what we are doing is we are now making everyone, it will take few minutes, everyone presenter, so that they can switch on their camera and mics, and then we can click a picture together.
Sure, sure.
Dr. Zishan, you can help me in doing that?
So this is request to everyone. In just 2 minutes, we will be able to enable the camera and mic of everyone. So please switch on the camera so that we can have a good group photograph.
I have changed the setting. They can turn on the.
Now it can be switched on.
Zishan, what we can do is you can just remove us from the spotlight so that everyone can come on the joint mode.
Yeah.
Student volunteers, you can also click the pictures.
Yes, all the participants can take pictures for themselves.
Everyone kindly adjust your camera so that your face can be covered in pictures. I request to every participant, so kindly they can switch on their cameras.
It's very nice to have
I have clicked a photograph in a together mode, so almost everyone is coming here. Thank you. So shall we conclude the session for the today?
Yes, we can end the meeting.
Thank you, everyone.
Thank you, participants.
And please, participants, don't forget